In this video, we are going to explore the hierarchy of protein structures using PYMOL. First, we're going to look at areas that have repeated patterns in the backbone. These are areas that are known to us as secondary structures. So we're going to start by revisiting the same protein that we looked at in the intro videos. We're going to look at the hemoglobin molecule, specifically the beta globin polypeptide chain. So once again, we're going to fetch 1VRE, that's the name of our file. Here's our file that we need. We are going to use a preset. So action preset pretty. And then we want to color by secondary structure. So we're able to color by secondary structure by going to color by SS. And I'm going to choose helix, sheet, and loop as a specific set. Aha. So here is our beta globin molecule. How many alpha helices can we see or can we count in the secondary structure of this molecule? So I'll spin it around a little bit. I see 7 secondary structures called alpha helices in this molecule. Now we're going to load a new protein because, as you can see, there's no other secondary structures in this molecule other than alpha helices. So we're going to load a new protein. I'm going to delete all, and I'm going to fetch the protein known as blue fluorescent protein. This is a variant of a protein found in jellyfish, and it's the name of the file is 1BFP. Aha! Here we have a very different looking protein than the one that we had before. But we're going to do the same thing where we use the preset pretty. And then we're going to color by secondary structure, and we're going to color the same way we did before. Now, what do you notice about this protein? Well, we do have some alpha helice turns, few of them. That we notice. The more dominant structure is these yellow arrows. Using our display sequence, we can click on parts of the protein to figure out which direction our beta sheets, these yellow arrows, point in. So you can see this arrow points in this direction to the end of the molecule. You can see that this arrow points in this direction, and if you were to sort of follow that, that also points to the end of the molecule. But we want to know if this end that we're examining here is our N-terminus or our C-terminus. So I'm going to click on the amino acid that's found there, which highlights the amino acid that's found there. And I wonder, okay, which amino acid did I select? Was it the one at the end of the molecule? Or was it the one at the beginning of the molecule? Well, we can see Met, which is our starting molecule. Met's over here. So in this case, we're actually looking at the very end, the C-terminus of the molecule. That's isoleucine right there. And our arrows point in that direction. We can also select a random arrow in the middle of our molecule to confirm that. I'm going to do this one. I'm going to select this amino acid. This is amino acid number 92, tyrosine. And then I'm going to select this amino acid, and you can see that gets me to amino acid 100, which is phenylalanine. So the arrow is indeed pointing towards the C-terminus of the molecule. So would you classify, knowing that information, would you classify the beta sheets as parallel or anti-parallel? Sort of think of what you would classify these sheets as. Okay, so we've looked at a couple of secondary structures. We looked at alpha helices and we looked at beta sheets. Now we want to look at overall tertiary structures. So we're going to go back to fetch 1VRE. We're going to go back to our beta globin molecule. We're going to change our representation to space filling. So we're going to go to show as spheres. And we can use the command line to select a specific group of amino acids. And we're going to name them small. So these are the amino acids that are sort of the smallest amino acids on the amino acid chart. And I'll just show you what that looks like. So in order to name the smallest molecules, smallest amino acids into one group, I'm going to write select small, so that is the name that I want, comma, and then I'm going to name the actual amino acids, and I'm going to name them by residue name, or RESN. And our smallest amino acids are glycine, alanine, and valine. And you can look those up. If you look at your amino acid chart, you would see that glycine, alanine, and valine are all very small. So I'm going to select them specifically. And as you can see on the molecule, those amino acids have been selected. And here, every alanine, every valine, and every glycine have been selected. And now I'm going to specifically color just those molecules. So I'm going to color green. And as you can see, only our smallest amino acids, the 3 that are the smallest, have been colored. So this is just an example. What you're going to do next is you're going to investigate the role of hydrophobic interactions in determining the overall shape of the soluble protein. So what we want to do is we want to color the amino acids according to the properties of the side chain, whether they're hydrophobic or whether they're hydrophilic. So we want to pretty much do the same thing that we did to create this group of smalls, but we're going to have a lot more amino acids because, you know, there are more hydrophobic amino acids in one group than just 3. So let's do the non-polar amino acids first. So I'm going to select, I'm going to name this group non-polar, and I'm going to type in all of the residue names of the non-polar amino acids. Well, if you were to look at your amino acid chart, all of the polar molecules are going to be either charged, right, or have nitrogen or oxygen groups that make them somewhat polar. And our nonpolar group on the right here has basically hydrogens and carbons. Proline and tryptophan sometimes throw people off because there is nitrogen in them, but for the most part they're largely nonpolar.
So I will take all of those amino acids that I just decided were non-polar. I will type them into the command line. Again, separating them only by plus signs. There should be 10. And I'm going to show, or I'm going to color those as yellow. So now I have selected and colored all of the nonpolar amino acids as yellow, and you can see sort of where all of those amino acids are found. I'm going to deselect everything, and I'm going to set a new group, right? And our new group is going to select the polar, actually I'll say select hydrophilic amino acids. Again, comma, open parentheses, residue names. We'll go back to our chart, check again to see where our hydrophilic molecules are. They should be on the left-hand side of the chart. And type in all 10 names of those amino acids into the command line. Again, separated by plus signs. Then I will close parentheses. Yep, that's the last one. Close parentheses. And I'm going to color by, let's do magenta. So I have yellow and purple. And so you can see, our purple molecules, our hydrophilic ones, are close to the outside. This makes sense because they are attracted to the aqueous solution that the molecule would be dissolved in. And our non-polar molecule, amino acids, are sort of found in the center, right, closer to the middle, and you can sort of slice through and see that they're found more primarily in the center, where they make LDFs and sort of attract to each other, as opposed to the water molecules nearby.
So hydrogen bonding is just one type of bonds that make up tertiary structure. We also see LDFs. We also see specialized covalent bonds called disulfide bridges between molecules that contain sulfur, amino acids that contain sulfur. And we also see ionic bonds. So I want to show ionic bonds in our molecule. So I'm going to go to a new preset. I'm going to return to simple. This is the preset we started with the very beginning of our activity on PYMOL. And I want to create a selection of amino acids that are acidic. And I also then will create a set of amino acids that negatively charged ions and our basic or positively charged ions should interact. And so we're going to make two groups, and we're going to differentiate their colors to make them easier to tell apart. So we're going to do the same thing that we did for hydrophobic or hydrophilic groups, nonpolar or polar groups, in sort of exactly the same way. So I would type in select acidic, comma, residue names. And I'm going to need to determine what are the residue names that are acidic. So if I go back to my chart, I'm going to see that aspartate and glutamic acid are both acidic. So their names are ASP and GLU. All right, so I've identified all of our acidic group. I'm going to show those so that we can see them. I'm going to show them as spheres, and I'm going to color those red. So all of our acidic side chains are now covered in red. In theory, these should interact with our basic side chains. So let's see if that theory holds true, or that idea holds true. So we're going to select basic, and now we need to type in the residue names that are in fact basic. So again, I'm going to go back to my amino acid chart, and I'm going to search for my basic amino acids, and I see that the positive charge is found on lysine, arginine, and histidine. So LYS, ARG, and HIS. Now I've got those selected. I'm going to also show them as spheres, and I'm going to color them blue. So I want to find a place where two molecules are closely interacting. I think I'm going to choose these two right here. I'm going to figure out what these two molecules actually are. So here we have an aspartic acid interacting with this molecule, which is lysine. Let's see if we can find another example. Okay, these two right here are close interaction, as we can see. But interestingly enough, they're not right next to each other. So we have arginine, which is way over here, number 117, and then we have aspartic acid, 21. So even though they're not close to each other on the primary sequence, they are interacting together with some sort of tertiary ionic bonding. So it's a really good example of ionic bonds in place. And you could draw the line angle structure of those two molecules interact. In the very last part, we're going to examine proteins that are composed of more than one polypeptide chain. Because we know that separate chains can come together to make a larger structure, usually through IMFs, that result in a quaternary structure. And so we're going to look at a couple different proteins that have quaternary structure. And the first is we're going to examine not just a single molecule of globulin like we've got here, but actual hemoglobin, the entire hemoglobin molecule. So I'm going to delete all, and I'm going to fetch 2-HHB. So this is a hemoglobin molecule. Similar in structure to the structure we've looked at before, but a lot of more going on, right? More complexity happening overall in the molecule. You can see heme structures, right? Those heme structures that we talked about in the last molecule are present, and there's 4 of them. So, think about what that might hint at or what that might imply. How many distinct polypeptide chains are present in the quaternary structure of hemoglobin? We're going to first put it into a simple preset. Aha. And if yours doesn't pop up automatically like this, what you should do is go to color by chain. And that color will appear so that you can count that there's a yellow, purple, blue, and a green, indicating that there are 4 globulin molecules in a large overall structure of hemoglobin. We want to list the first 6 amino acids of every chain, and so you can do that up here by seeing that the chains are color coordinated. So I'm going to go ahead and select the first 6 of my green. I'll go ahead and select and then finally I'm going to go to my yellow, val, his, lu, threonine, proline, glutamine. Okay, this also looks familiar with me. If I go back to my blue, starting val, his, lu, threonine, proline, glutamine. So I'm noticing that there are in fact shared characteristics between blue and yellow and between purple and green. So use this to explain why hemoglobin is described as what we call a heterodimeric protein. So, we have a complex quaternary structure here. We have 4 chains, but we're going to look at an even more complex quaternary structure. So I'm going to delete all, and I'm going to fetch a new molecule. This is a protein from Staphylococcus bacteria called hemolysin, 7-AHL. It may take a little bit to render this molecule because this molecule is just so incredibly large. So if it takes a moment to render on your computer, don't be surprised. And then you can see this is a very, very large protein. We are going to go back to preset. Simple, in this case, no solvent. And then if you aren't colored by chains, please make sure that you go to color by chains. And you should be able to count now the number of chains that are here. 1, 2, 3, 4, 5, 6, 7 separate polypeptide chains that make up the quaternary structure of this molecule. We can measure the diameter of the pore in the middle of the molecule using a measurement wizard. So this is pretty cool. Feature where you click on an atom on one side and an atom on the other side, and you try that. That's a little bit better. So that one says 24.1. Maybe we'll try this and this. It says 31.5. This and this. Hard to get a perfect example, but somewhere between 25 and 32 is sort of our measurement. And what unit of measurement is this? This should be in angstroms. I'm going to do one more thing with this image. I'm going to show as surface. I'm curious sort of what this protein looks like. Again, it's going to take a little while to render, But now we can see the protein, we can see the pore in the middle, and we can sort of determine overall what this would look like in a molecule, in the cell, what this molecule would look like in the cell. So the protein is thought of a mushroom shape. What kinds of amino acids are found in the outer surface versus, of the cap, this part right here, versus the outer surface of the stem. And I'll tell you that this molecule is embedded in a membrane structure, with part of the structure of the molecule hanging out of the membrane and part of it embedded in the membrane. So that might give you a hint as to the type of amino acids that you find in both